I describe myself in minor poetic terms as a artist stuck in the body of a science writer. And as such, I prepared a presentation, which I call for you, Beetlemania Has Not Bitten the Dust. And you can consider it a pre-Halloween address, as we'll see. As Paul alluded to, I think my mother, Lynn Margulis, could have won 2 Nobel Prizes, one in physiology or medicine for symbiogenetics and its relationship to health in the long run, as well as for the Gaia hypothesis, Earth as a geophysiological system that regulates major aspects of the atmosphere, marine, and of course, the soil environments. She did not, as we also heard, discover symbiogenesis as a phenomenon. There was Mereshkovsky and others, Ivan Wallen in the United States, especially, who had put it forth before, but it was her interdisciplinary scientific work and nonstop energy supplied, by the way, by her mitochondria, also the same mitochondria that are powering this talk because they're matrilineally descended. So her mitochondria are powering this talk through her exile, even though she is no longer alive. And all that together work to get symbiogenesis accepted. As mathematician Augustus Morgan wrote in 1872, Book of Paradoxes, appropriately stealing a bit of poetic code like a virus or a bacterium from Jonathan Swift's 1733 poem on poetry, a rhapsody, quote, "Great fleas have little fleas upon their backs to bite 'em, and little fleas have lesser fleas, and so ad infinitum. And the great fleas themselves, in turn, have greater fleas to go on, while these again have greater still, and so on." As contemporary symbiosis expert, actually an animal physiologist and biochemist, Margaret McFall-Nagai of the Pacific Biosciences Research Program at the University of Hawaii in Manoa points out, we are colonized by very specific microbiota. And the bacteria in the crook of your elbow, beneath your arms, at your groin, and in your toes will, like European colonizers settling places similar to their homes in the old country, be more like those at those places in another person's body than like other microorganisms elsewhere on his or her body or your own. On our skin, inside our bodies, in the evolutionary history of our animal cells, are known and unknown life forms, some of them earning their keep evolutionarily, or at least not spoiling the party for everybody. Some of their genes find their way into our genome. Over time, they become us. McFall-Nagai also pointed out, appropriately to a room full of physicians, that in the lush marine environment of our ancestors, with 100,000 cells of microbes per cubic centimeter, 100 million per liter, animal immune systems may have evolved not so much to keep out others like border patrol agents, but to welcome in select neighbors whose diverse talents were co-opted for the common good. We see this, in fact, with the luminescent bacteria functioning brilliantly in the organism upon which McFall-Nagai is a world expert, the Hawaiian bobtail squid, Upramina scalopes, which has a special light organ, replete with a lens like its own eyes that focuses the energy produced by its permanent road crew, the teeming luminescent Vibrio fischeri, keeping them happy with a continuous open bar of protein drinks, that is, amino acids and sugars. So they can, in effect, party forever, bobbing pleasantly up and down, largely free from harm between the deep blue sea and the starry night above the Pacific Ocean. Largely free from harm because of a process called counter-illumination. The bacterial light reflected off the squid's reflector on the underside of its body and focused by the lens of its light organ downwards adjusts to mimic the ambient light of the stars or the moon, even when it's full. In this way, potential predators looking up don't see the tasty silhouette of a cephalopod sea creature. They see nothing but the night's light, which allows the bobtail squid to hide in plain sight. It is worth noting too that the bacteria and the cephalopod, although belonging to different kingdoms and domains of organisms, are more than just friends. Squid do not develop to maturity without Vibrio fischeri, dinner guests who find the sauce so good that they are in no hurry to leave, in effect, marrying the cook for his or her continuous buffet. Well, this bright example gives us a kind of evolutionary window or snapshot between symbiosis, the living together of organisms for the greater part of their life cycles, and symbiogenesis, the living together of organisms until they can no longer do without each other and become new beings, including new species and higher taxa of life. This is the case with our cell ancestors and the organisms that invaded or were devoured by them, becoming mitochondria. In symbiogenesis, organisms come together forever, something that the squid and its bacteria seem to be doing. And in a living system where one's needs are taken care of, such as are the Vibrio fischeris by the squid, you can eventually lose parts of yourself. At the limit, as Sir David Smith put it, all that may remain of the hosted organisms are mysterious elements, like the Cheshire Cat's famous grin in the Alice in Wonderland. When we look at life from a symbiotic onto symbiogenetic perspective, it becomes clear that the characterization of viruses and bacteria as germs to be avoided, the still prevailing medical perspective, is only a small part of the story. If you destroy all your hosts or ruin your living environment, you yourself and any special genes you carry may perish. In this sense, more, not less bacteria, more, not less fungi, protists, and archaea may be a better safeguard of health than misguided and ultimately impossible ideas of purity. I am as pure as the driven slush, quipped the daughter of an Alabama senator, Tallulah Bankhead. In classical ecology, as well as the strangely parallel situation, ontogeny, the growth of embryos, there is a tendency for greater energy capture, greater biodiversity, and less rapid growth over time. Early ecosystem development may be marked by rapid colonization by quickly reproducing settler species, but over time, their growth slows, biodiversity increases, and metabolic variety necessary to recycle waste and close or nearly close local biogeochemical cycles arises. Thus, in retrospect, from a more Gaian symbiogenetic perspective, the germ theory of disease, important as it was at its time, definitely better than the demon and devil theory of disease, to which it nonetheless bears a family resemblance, is a very partial perspective. What we call disease is in some cases roughly equivalent to the unequal growth of one or several species or kinds of life at the expense of others. It may even be worth suggesting that the rampant growths are not so much the cause of disease as a lack of requisite diversity at both the ecosystem level and individual level. Consider, for example, Candida albicans, the cause of thrush, diaper rash, yeast infections, and parleche, the cracked red corners of lips. A normal cohabitant of the human body, this fungus is one of the first to devour a corpse. Health at the body level mirrors that of an ecosystem at a larger level, with the fast growth abating, transitioning to a more steady turnover of matter, one which avoids depleting local energy and matter sources by overuse, which would entail an overproduction of waste, including the final heat waste of entropy. Too much waste, including heat too close to a population's or ecosystems or biospheres' sensitive working surfaces, will impair, sometimes fatally, its ability to function as a natural thermodynamic system. A parallelism thus theoretically exists at the synchronic ecosystem and diachronic evolutionary levels. Ecosystems' health related to biodiversity, steady energy flow, and successful transportation and transformation of waste is mirrored by the sometimes multi-metabolic systems evolved via symbiogenesis. Here we may consider plants superior life forms, as they both respire, as we do, using their mitochondria, but also photosynthesize, using their cyanobacteria-derived chloroplasts, making food for themselves and others from water, starlight, and air. As some of you will know in far more detail than I, the soil as a skin of earth consists of many organisms. We can imagine early life, whether endogenous or comet delivered, arriving or quickly evolving to hardy archaean and bacterial forms that availed themselves of gene trading, were swimming in genes due to the lack of ozone layer that they themselves would later produce in the initial oxygen crisis of bacterial evolution, and so on. The pressure of spreading life and the advantages of multiply evolved forms of metabolism of bacteria allowed them to cover the planet with successful long-term ecosystems. With algae and fungi, the land was covered as atmospheric gas composition stabilized into a continuously reactive but metastable condition, a gestalt reflection of the homeostatic abilities of evolving, reproducing, gene trading, waste-producing bacterial cells. Within this planetary maelstrom, symbiogenetic organisms played their part. The alpha proteobacteria, similar to rickettsia, which may have changed the outcome of some wars more than armies as typhus, were champions of the post-Holocaust world of oxidized metals, tissues, and beings in a newly oxygenic atmosphere that was largely the legacy of the fast-growing cyanobacteria. Having mutated or otherwise evolved, the cyanobacteria tapped into an immense raw material for their photosynthesis. Bonds broken, the hydrogen atoms in water became available as electron donors in photosynthesis. In this new environment, alpha proteobacteria and other oxygen respirers flourished. As with the rapid spread of monocultures over a fresh corpse, or rapidly growing pioneer species early in ecosystem succession, the alphas tended to invade and attempt to devour, or what is the same thing from the inside, infect their juicy targets. And in those many tens or hundreds of millions of years that oxygen was accumulating in our newly blue atmosphere, the old guard of anaerobes that once ruled the surface had to go underground, like mythical beings in a Celtic fairy tale. Either that or stay out of trouble, harm's way, by taking refuge in other beings. This is what the anaerobic archaean beings known as methanogens eventually did. And they now, from their oxygenless bunkers inside the rumens of cows and the hindguts of termites, produce globally significant amounts of methane into our atmosphere. Our eukaryotic ancestors didn't like this either-or choice. Recklessly, they refused either to bury their heads in the muds or find a suitable, not-so-temporary bunker until animals' digestive tracts finally evolved. It's one thing to hold your breath for a few minutes or wait until the age of maturity to marry your dream beau. It's another to trust your luck to the gods for 1,000 million years. Still, the archaean anaerobic hosts, some of them anyway, received the incredible stroke of luck they were probably too brainless at the time to bank on. Happily, some of them became afflicted with a killer infection that ended up saving them. Our body cells, like those of plants and fungi, and most protoctus, protists, slime molds, protozoa, kelp, for that matter, had ancestors who testified to diversity's triumph long before man, indeed of which man, from a certain perspective, is only a pointillistic variation within a huge mural of shimmering gene-trading microbial activity. Animals are clonal and often sexually reproducing multi-tissue beings, developing each generation from fertilized eggs with two sets of chromosomes that initially grow exponentially like ecosystems or bacterial cultures in a petri plate before they run out of agar. In each, almost all, of the body cells of such beings, we find mitochondria, transducing energy and making ATP, adenosine triphosphate, the energy currency of all cells. Mitochondria with genetic sequences, morphology, size, metabolism, and reproductive lifestyles tying them to respiring bacteria. Lynn Margulis's theory of the origin of eukaryotic cells by serial endosymbiosis refers to the composite evolution of so-called higher life forms from aggregate bacterial colonies. In the case of mitochondria, she hypothesized their valiant ancestors invaded and interpenetrated, infected, if you like, the anaerobic ancestors of eukaryotic cells struggling at the surface of an increasingly oxidizing planet. The most important later addition were the cyanobacteria, who had created the mess in the first place. Among these were the ancestors of modern forms of algae and brown and green seaweeds, as well as plants, which are distinct in that they developed from sexually reproduced embryos, which we like to consider frozen ecosystems that had moved to a macroscopic domain. Once heretical views of the bacterial origin of mitochondria that produce the weight of your entire body in ATP each day, and the green parts of plant cells which make themselves and provide food for the planet in effect like magicians from nothing but water, air, and sunshine, are now taught as a matter of fact in textbooks. Cyanobacteria own the means of production, supporting the entire human economy such as it is, as well as the far larger, older, and stronger planetary ecosystem. She also believed there was another, more mysterious being deeply involved in the melange that became the eukaryotic cell. This was the final part of her serial endosymbiosis theory she was still working on at the time of her death. I have been focusing on symbiogenesis in the epic events that led to the basic outlines of our and other eukaryotic phyla, but there are many other fascinating examples, such as the role of Wolbachia bacteria in insect evolution. Members of the genus have been electron microscopically photographed curled up inside the germ cells of insects, 70% of which, by some estimates, are infected by them. In some species, they change the insect's sex or are considered indispensable to their health, similar to our own microbiomes. Wolbachia infections coordinate with female-only populations of wasps, and these bacteria have been found in mammals as well. When the British-born geneticist and math whiz, later Indian citizen J.B.S. Haldane, who basically originated neo-Darwinism but didn't necessarily ascribe to it, a Marxist who, like his father, used himself as a guinea pig for biomedical chemistry experiments at home, and who learned to read at age 3, when Haldane was asked if he learned anything from his many years of study about the nature of God, he quipped, "He has an inordinate fondness for beetles." You should think of this story when you look at a beautiful ladybug, aphid-eating coleopteran arthropods of the family Coccinellidae, considered good luck and a sign of ecological health. My prolific mother, with whom I worked closely for 30 years, near the end of her life, in a debate at Balliol College of Oxford University, moderated by Richard Dawkins' advisor, Dennis Noble, responded to the famous science popularizer that when Haldane made his famous comment, he should have completed his sentence. The full sentence, spoken by the man who invented the term primordial soup, as well as some of the foundational ideas of neo-Darwinism, should read, "God has an inordinate fondness for beetles and their Wolbachia symbionts." What do I mean? She continues, addressing Dawkins, who conceded that symbiosis may have played an important role early on in the evolution of eukaryotes, but plays little, if any, role in ongoing animal evolution. She explains to the zoologist, who does not seem happy about microbiology encroaching into his hallowed animal domain, they've found spectacular cases where differences in Wolbachia, which you don't see, they all look like beetles. You don't see them because you don't see those bacteria. Nitrogen metabolism differs in the Wolbachia. Hormone production that changes gender of the insect differs in these Wolbachia, and all female wasp populations are associated with Wolbachia infections. But the coup d'etat was, and this is her speaking again, but the coup d'etat was when the entire Wolbachia genome was sequenced, and it's not even the size of typical bacteria, it's smaller. The genome was labeled by hybridization DNA, and the whole damn genome of the Wolbachia is on one of the autosomal chromosomes. The fact that the entire genome of that bacterium has been incorporated into the chromosomes of the insect nucleus, how much more intimate can you get? So it's genome acquisition, it's not gene mutation. Now that's symbiogenesis. It gives a speculative answer to the question implied in atheistic Haldane's provocative answer. Nature's inordinate fondness for coleopterans, the beetles, is because of no music or magic more mysterious than the ancient, one might say Ovidian, power of symbiogenesis. Which brings us full circle back to my beginning comments of bugs inside bugs and a living lamp, which is the bobtail squid, and the nonstop rave its protein drinks provide for its bright band of bacteria. Camouflaged by bacterial light from predators, produced in their light organ, bobtail squid blend their own light to match that of the stars or the moon. Wolbachia, with its minuscule genome, is a mycoplasma. It's among the smallest and genomically minimal bacterial cells. In the Hawaiian squid, they still live on their own, but they eventually may become one organism. In a biosphere's living environment, one can lose parts of herself, blend. Killing infections can become needed organelles, the children of bitter enemies, closer than the closest friends. This is symbiogenesis, a major evolutionary process on a symbiotic planet. Wolbachia, known to transfer their genomes, are reminiscent of mycoplasmas, that corrects my earlier statement, minimal bacteria that don't even have a cell wall, with the smallest known cell genomes, perhaps a result of living so closely for so long with others. So too does Rickettsia prowazekii, an alpha proteobacterium, as were the ancestors of the cells that became the mitochondria in your cells. So do they too have a reduced genome. It does not code for all its own proteins, and it is carried by human body lice, tiny arthropods that can't even jump or fly, but have crawled for hundreds of millions of years before our ancestors walked. When it was sequenced in 2010, it won the contest for smallest insect genome. Such cases can probably be ascribed to the tendency of beings living in other beings to continue to survive in increasingly minimal, ultimately ghostly forms when they stochastically lose parts of themselves. The universe is not only stranger than we imagine, to paraphrase another bon mot of J.B.S. Haldane, it is stranger than we can imagine. This Halloween, we might think of that Cheshire smile that Sir David Smith of the Royal Society mentioned in his address on symbiosis, and as our full, even perhaps, and the idea that we are full, even perhaps made of such bacterial ghosts. Thank you.